Okay, so we are back with some wrap up here. We've done with meiosis, and these are a few little wrap up applicational concepts that go along with meiosis. All right, so let's go over the processes of gametogenesis with application. We actually have already done that because the creation of sex cells, ladies and gentlemen, is gametogenesis. All right, the process by which a diploid cell is going to undergo meiosis and give you a haploid gametes. All right, so we're going to show you both oogenesis and spermatogenesis in a couple of pre-drawn diagrams by me, which you are then going to draw into your Cornell notebook. Start making that list again of things that I want to draw in the Cornell rather than you go find pictures. I'm going to make sure you understand that. But let's go to the smartboard. Okay. I have pre-drawn, for time's sake, a diagram on spermatogenesis. Now, this is spermatogenesis in a human, but any organism, you take the number out, any organism can have. All right, legend goes that the SS means single-stranded and the DS means double-stranded. All right, now the diploid number in humans, as you know, is 46. That's what it is. Doesn't matter if it's single-stranded or double-stranded. You see the number 46 and it's diploid. However, in the germ cell, that's the precursor cell, there are 46 chromosomes. That 46 must duplicate once, so you have 46 double-stranded chromosomes or single to double, which are identical, okay? And identical chromosomes then are going to then start to separate. So the 46 single, 46 double, and the precursor, again, you're going to have multiple things in the cell, organelles needed for the sex cell. All right, so moving through, it goes through its first meiosis, and then it separates one, the 46 double-stranded, separate into 23 double-stranded. Once you see the number 23, that's a haploid cell. It doesn't matter whether it's single or whether it's double. 23 is the haploid number in humans. All right, so it separates ones, prophase, metaphase, anaphase, telophase, two cells. Those two cells are not sex cells. We need two meiotic divisions, 1 and 2, to produce 4 haploid daughter cells. Yes, they're called daughter cells, even in making a sperm cell, with their own unique DNA in it. Now, again, remember, it is unique because in meiosis 1, you have crossing over, so these chromosomes are probably not exactly the same going through. All right, so one big caveat when you're looking at meiosis 1 versus meiosis in a female is that you're going to have 4 sperm cells for every meiotic division, for every biotic division. Okay, 4 sperm cells. And you notice there, meiosis 1, meiosis 2. So this is to be compared again to the germ cell, 46 single-stranded back to 23 single-stranded. And you're going to put the single-stranded together with the single-stranded, and you're going to get a fertilized egg, we hope. Let's go to the egg. All right, same thing, but it looks a little different because remember, here's the big difference between meiosis. One egg cell. All right? All right, so the egg is waiting, all right? And during the menstrual cycle, as you saw during the follicular stage, the germ cells mature. Part of that maturity is going from single-stranded to double-stranded, all right, going through the process of meiosis, through the cycle to get ready for ovulation, which obviously you're going to need this down here. So same things occurring until you get to the end. All right. How does an egg make an egg? Remember, an egg cell is easily seen under a very low-powered microscope. All right. Why? Because the egg, you get an unequal distribution of cytoplasm. So you divide sperm cell, you go back, equal distribution of cytoplasm. Egg cell, unequal distribution of cytoplasm. And you create these smaller cells, which are all called polar bodies. Now remember here, this is going to divide again and give you another cell, which I'll make a little bigger here. which is the egg cell. This is the egg cell right here. All right. Egg cell, much more cytoplasm than any of the other polar bodies. One of the reasons why these can't develop into eggs, the polar bodies, is that they don't have enough food in them. Remember, an egg is an egg. An egg has the yolk, it has the white part. The white part is its proteins. The yellow part, food for the egg. And you only need to nourish this hopefully for about 48 hours or so. All right, so you see here, for what we want to know, 23 single-stranded and at least 3, sometimes 4 polar bodies, unfertilizable. All right, now sometimes you see the diagram, you don't even see these 3, you don't even see these 2 here, 2 polar bodies, but be averse to seeing a few diagrams. Let's take a look at what the PowerPoint says here. All right, so gametogenesis, we've gotten through there. This gives you another look at different picture, 4 chromosomes to 2. Okay, back to, all right, the single sperm. You are not responsible this year for spermatogonium or these scientific word. No, not this year. All right, so, wait. Oogenesis, all right, you want to go to the ovum. There you are, secondary.